Okay. Okay, right. So let's start over again. So previously we talked about the virus, and we discussed whether virus is classified as alive or non-alive by itself. So the question, the answer is no, right? Virus is not alive. Even it can replicate itself. Even it can be infectious. Um, the virus is not alive because it needs the host. It needs the host cells to help it to replicate, right? So it cannot replicate without the host, and there's no activities that the virus can do without the host. Okay, so what contains in the virus? Okay, so when we look at the structure of the virus, we can see that here. So we're gonna see that it has the DNA or RNA, which are the viral genomes, right? So these are the code that will allow virus to function. So allow virus basically to make copy of themselves to cause the disease, or to release and spread more of the infections. Okay, and those genomes are surrounded by the structure. It can be structural proteins like the capsid proteins, or some virus can be surrounded by the envelope, right? By the envelope, which is going to be the lipid bilayers. Okay, so those are those functions, the functions for the the structural proteins or the envelope is meant to protect the genome of the virus, because the genome of the virus is very valuable for the virus, right? So that's why it has to be protected. Okay, so here is basically the comparison between the cells, the eukaryotic cells, which is considered the basic living things, right? And the virus particle, you can see that the virus particle can contain only the genomes, right? And contain the structural proteins or the envelope. But in the eukaryotic cells, it contains like many organelles that can function, right? It can do many activities in the cells. Okay, so that's why that is what difference between the um, the cells and the virus particles. So the virus on Earth, right? Virus is very common. So they are over, right? They are over 10 to the 31 viruses. So you can see like how many they are on Earth, right? So, but not all of them can cause the disease. So most of the virus are not gonna cause the disease. So as we know that right now, in everyone's body, so we have like approximately 380 trillion viruses. So how large 3 trillion is? 10 to the 12, isn't it? Okay. So 10 to the 6 is million, right? So 10 to the 9 is billion. So trillion is 1,000 times a billion, isn't it? So 10 to the 12. Okay. Viruses in our bodies right now. So if they all are if they all cause the disease, we're gonna be dead by now, right? So, but some of the virus can cause the disease that and it's push a lot of pressure on the selection, right? Because so in the primitive time, so when the cells, or even when the animals, right, got infected, so if they cannot adapt, so they're gonna be die, right? So only the animals that can adapt it from the infections will be alive, right? So that is going to be another pressure for the selections. Okay, and what shape us to be us right now? One factor is the virus, right? Because our ancestor got infected by many kind of virus, and like some functions of our body might like derive from the infections in the primitive time. Okay, so that is what the virus infection. So the size of the virus. So the size of the virus, I have the scales here for the comparison. So we're starting from the size of atoms, which is 0.1 nanometers, all the way to the frog eggs right there. So that's what we can see by our eyes, which has size around 1 millimeters. So virus is in the middles right here. It's around the middles. So I have the flu virus here, which have the size around 100 nanometers. So which is not be able to visible by our eyes, right? Even with the microscope, with the light microscope, we still cannot see it, right? But larger than the size of the virus, for the size of bacteria or the plant cells, the animal cells, we can see it under the microscopes, right? But most of the virus, we cannot see it under the microscopes. And in terms of the genome size, the genome size here can be like diverse as well. So we have the genome starting from the small size of the viral genomes, like 100 kilobase pairs here, all the way to like it can be millions of base pairs. Okay, so that is the variety of the virus. So as I said, the virus can be seen under cannot be seen under the light microscope, but the tools that we can visualize it, we have to visualize it under the electron microscope, right? So I'm going to be talking about the electron microscope later when we get into the structure of the virus. But here is kind of like the example of the pictures after we capture from the cryo-EM or cryo-electromagnetic microscope. Right? No, cryo-electron microscope. Okay, so here is the viral particles. So the viral particles compared to the size of cells. So this is a cell, right? So this is a cell. So here's the picture of the virus when it's transported out of the cells. You're going to see that the small size of the particles here is the virus particle. So that is how small it is compared to the host compared to the host and compared to the size of the whole cells. And here is the viral structures, right? It contain the genomes, contain the capsid proteins, and some virus contain of the envelope, and some virus can also contains the spike protein as well here, which is responsible for the attached to the cell's receptor. Okay. Okay, so let's go to the viral genome here. So the viral genome here is very important scheme. So we call it Baltimore scheme. So Baltimore is the scientist who got the Nobel Prize from studying the mRNA, right? Because that's why he put the mRNA here in the center. Because mRNA is kind of like the intermediate of the protein in the protein synthesis, right? So if we have DNA has to be translated to mRNA first in order to make the proteins. And all the virus want to make the proteins because the proteins can do the functions, right? Protein can do something to the virus. Also, proteins can make the structure of the virus too, because the structure of the virus will contains of the proteins. So all the virus can want to make the mRNA first, then to synthesize the proteins using the mRNA. So if we classify it from the genome of the virus, we can classify them to 7 types of the virus. Okay, so let's start with the double-stranded RNA, double-stranded DNA first, which is one of the common type of the virus. So if the virus contain DNA, right? So when they get into the cells, DNA needs to be translate to needs to be transcribed into mRNA first, right? In order to make the functional proteins of the virus, then it can do replications, or it can like assembly the virus and make lots of virus particles and export out of the cells. Okay, so that is the DNA. And here on top right here is the single-stranded DNA. So some virus have the single-stranded RNA as the genome, right? So the single-stranded RNA here cannot be made to mRNA directly, right? Because in order to make mRNA, so we're gonna have to make DNA first. Okay, so that's what we learned in the transcriptions, right? So it has to make the complement of this DNA, right? In order to make the double-stranded RNA, double-stranded DNA. Then after that, it's gonna make the mRNA. Okay, so there's gonna be the 2 steps of the synthesis for this virus. So the virus that contains single-stranded DNA will kind of like have slower, right? Slower for the process, the whole process. I mean, including the infections and replications and everything, because it needs a lot more steps. Right? It needs one more steps to make the mRNA and then make proteins compared to the DNA. Okay, so and I have a sign right here. A sign right here means the sense of the genome, right? So let's start with the mRNA here. So mRNA, because it is the single-stranded RNA, right? So we can call it the positive sense. The positive sense, it doesn't mean that it contains the positive charge and anything to the mRNA, but the positive sense means the right copy, right? The right copy. So if we have the positive, right, we're gonna have the negative. So the negative actually means the complement of the positive sense, right? So we're gonna call this the negative sense. Okay, so because in order to make the proteins, we need the single-stranded RNA, right? So the complement to that single-stranded RNA is going to be the negative sense or the negative RNA. Okay, so I label it as the olive here, and the green here means the mRNA, the positive sense of RNA. Okay, so that's also applied to the DNA. So we have the positive sense of DNA. So in order to replicate or in order to transcribe to mRNA, it has to make the complete DNA first, or double-stranded DNA. Okay, and then let's move on to another type of DNA here. So we have the gap DNA. All right, so the gap DNA here, even it contains the positive and the negative DNA, positive sense or negative sense DNA, but it cannot be transcribed to mRNA, because in cells, when it found the damage or gapped DNA, it's gonna have to repair it first. So this type of the virus, if it has the genome, the gap DNA is gonna have to repair the DNA first to make the double-stranded DNA and then like transcribe to mRNA. Okay, and then we will see the next type, which is the double-stranded RNA. So this one is also like exists as well. So for the double-stranded RNA, again, it cannot like transcribe directly to mRNA. So because this RNA, right, it has the double-stranded. So those double-stranded has to be separated first, right, and then in order to make the mRNA. So then we talk about the negative sense of RNA a little bit. For the negative sense, what it has to do is has to synthesize the complement, right? The complement of this sense of RNA first. So that complement of negative sense RNA is going to be the positive sense or mRNA that we need. Okay, and then let's talk about the left side right here. So left side here, they both actually contains of the positive sense of RNA. So this type, even they contains the positive sense of the RNA, they're gonna be like reverse transcribe to DNA first. All right, so there's going to be the additional steps that the virus can do. So there's like some particular groups of the virus that actually have to do these steps. So I'm going to be talking about it when we learn about the this type of the virus. So after that DNA will be made, right? And then double-stranded DNA will be made, and then after that, it's going to be positive sense of mRNA. Okay, and then another type is going to be positive sense of RNA that will make negative sense of RNA first, right? Do you know why it has to make the negative sense of RNA? So because yes, that's correct too. And but another thing that they want to make the negative sense is they can replicate, right? So they can replicate using the negative sense of RNA, and then the negative sense of RNA can also convert, or I mean, like synthesize the positive sense of mRNA, which is going to be like the correct complements of the mRNA that we need. So those are the seven types of the viral genome. So all of them, right? All of them, you're going to see that the arrows point into positive sense mRNA. Okay, so that is importance. Even we have like, even this one, right? This one, we see that it's want to synthesize the negative sense of RNA, but this one, the positive sense of RNA itself, it can transcribe the proteins too. So we can we're gonna see like later in the example about like the type of the positive sense of the RNA in the genome. So it's gonna have like some different characteristics from other virus. Okay. All right. So let's move on to the next one here. So we classify the viral genomes, right, by the types of the genome, whether they are DNA or RNA, whether they are double-stranded or single-stranded. But another thing that we can classify the viral genome is by the structures. So the structure of the genome in the virus uh, can be various as well. So we can have like starting from linear, circular, segments, gapped, right? And then so we have like stranded here, single-stranded, double-stranded, or cross-linked. So those are kind of like the diversity of the structure of the virus genome. Okay. So another question is like, let me go back to here a little bit. So So we may have a question that why do viruses need to have like this such variety of the genomes? Why don't the virus only like contains mRNA? Because mRNA is the most important, right? It's the most important for the protein synthesis. So why don't all virus just contain mRNA? So that may be a question, right? So do you want to like guess why? Yes, yes, that's correct. So that is that is what the what make the it's kind of like the pressures, right, to make the virus um reverse transcriptase to DNA. So and some of those virus stores its genome in DNA instead of RNA because DNA is much more stable, right, compared to mRNA, right? And I and another thing that I want to show you here. So you see the differences between the size of the genomes here. So here on top, I put the largest virus in terms of its genome. So we have the Pandora virus here, which contains the DNA, right? So this virus has 2.4 million base pairs, right? So these are ones of the largest genome for the virus. But here, the largest genome for the RNA, you're gonna see like it's the different stories compared to DNA, right? So the largest RNA virus contains only 41 kilobase pairs, right? Another thing from having the DNA as genome is the capacity, right? The capacity of like having the informations contained in the genomes, because again it's point back to the stability of the DNA over the RNA, and then DNA can be like much longer than the RNA, and that's why it can contain a lot more information, okay, compared to RNA. And then like we go all the way to the one of the smallest virus, circle virus here, which has only 0.8 kilobase pairs, right? Which classified as the RNA virus. Okay, so we can see here the name is very funny. We have like Mama virus, we have Mimi virus, and then we have like Momo virus. So these are kind of like the series of the virus. So those are classified into the same family, and those family have like a very large size of the genomes. Okay, so let's move on here. So in the viral genome, what it contains in the viral genomes, right? So in the viral genomes, each regions will transcribe the different kind of proteins, and those proteins can be either the structural proteins or non-structural proteins. All right. So for the structural proteins, it will be included as the structural of the virus. So it can be like the capsid proteins, right? It can be the envelope proteins, or other than that, for the non-structural proteins, they are going to be the proteins that do some functions in the viral replications, right? Or some of the non-structural proteins will cause the disease. It will like, for example, suppress the immune system of the host cells, right? In order to allow virus to replicate more efficiently. Okay, so like for like this example, I think I have the the viral genome of hepatitis C. Okay, so in hepatitis C, they're gonna include the protease here, and then the polymerase and the helicase, right? So those have like pretty much important functions to assist the virus to make copies of themselves, right? Like for example, for the protease will help to clip and activate those structural proteins and non-structural proteins, right? Because when virus get into the cells, it's gonna transcribe the whole proteins right here. So we call it polyproteins, right? And in order to activate each protein, it will need either the host or its own protease. So that's why it's want to like synthesize its own protease as well to make the replication faster and more efficient. And then like some of the proteins right here, like NS5, is the polymerase. So virus can um, can can transcribe the proteins that actually existed to make more copy of itself, right? So you're gonna see that in when the cells get infected, even like just the small numbers of the virus particle, it can make like 1,000 times of the virus. Okay, so okay, let's start with the double-stranded DNA. So this one, it's more like more familiar, right? We're gonna be familiar more with this type of virus because it contains double-stranded DNA. So we noticed that very well that the DNA need to be transcribed to mRNA, and then our mRNA will make the proteins, right? And then the proteins fold, and then those folded proteins will like get together, assemble together, and make the new virus. All right. So this process happen over and over to make numbers of the virus. So the example of the virus that contain double-stranded DNA here is the polyoma virus. So polyoma virus is the virus that infect in our renal organs, right, or internal organs. Right. So it's one kind of the virus which we call the chronic infections, right? Once we get the infect, it will stay with us for like years, can be 10 years or 15 to 20 years. But when our body is weak, it's gonna wake up, right? And it's gonna cause some damage to us, all right? Okay, so here's other types of the double-stranded DNA. So we have like adenovirus, we have herpes, herpes viridae, which can cause kind of like can cause the herpes, right? Or the skin damage, right? After we get the infection. So this one can spread to the body contact. All right, so or the skin contact. Okay, and then next is the papilloma virus. So papilloma virus. So the one that we kind of like more familiar is HPV, right? Or human papilloma virus that can cause the cervical cancer, right? It can cause the cervical cancer with after we get the infections. Again, so this kind of virus is kind of like the chronic virus, and so because that's why it's recommended for everyone, right, to get a vaccine. You may heard that HPV vaccine, especially for females, right, because that can cause the cervical cancers, and polyoma viridae, that what I already talked about, and pox viridae. Okay, so this one basically from its names, right, it can cause the like some pox, and then we have like a huge structures right here, pitovirus. So the size here, it is very large compared to other kind of virus. So now one of the reason why it can be this huge, because it contains the double-stranded DNA, right? Because the double-stranded DNA is very stable, so that's why it can like have like the huge structures of the virus for this type. Okay, and then I'm gonna go next to the gapped double-stranded DNA. So for the gap double-stranded DNA, so it will need additional steps to the double-stranded DNA in order to make the mRNA. So this is mRNA, All right. So for the gap or the truncated type of double-stranded DNA, when it get into the body, so the body will like trigger the repair system. So they will like sense that this DNA is damaged. So it's actually like depends on the cell's machinery to recognize the damage and then repair the like the gap DNA first, right? And then to make the complete double-stranded DNA before transcribe to mRNA or the positive sense of the mRNA. All right, so it can be used in two purposes, right? The first purpose is like obviously mRNA, the positive sense of mRNA is going to be made, it's going to use to make the proteins, right? And then proteins can be used to assembly into the new structure of the virus, or it can be used as the non-structural virus. And then another thing that it can do is gonna reverse transcriptase or reverse transcribe to a minus strand of the DNA, right? So minus strand of the DNA can be polymerized to gap DNA, right? Which is the original genomes of this type of the virus. All right, so in order to make the new virus, it needs to make make a lot of copies of the original genomes too, right? So that's why it kind of like also depends on the reverse transcriptase to convert the RNA to DNA, and then from DNA to the gap DNA here, and then put together with the capsid proteins and all the structural proteins, it will like assembly to make the new virus here. So the example of the virus in this type is the Hepatnoviridae, sorry, which is the one that I think it's gonna cause like the disease in kind of like more all the mammalians, right? Not for the humans, but for hepatitis B is right here. So from its name, right, is the virus that can infect and stays in our liver, right? It can cause also the chronic disease like if it stay in our body for a long time, it may cause the cirrhosis and liver cancer, right? Eventually. Okay, so this is one type of the hepatitis B. All right, if actually hepatitis virus, there are a lot more hepatitis virus. So you may hear that hepatitis A, right? Hepatitis C, and so on, right? So I think there are more than 5 types of hepatitis virus, but each of them, right, have different genomes. Like for example, we have hepatitis B virus that have the gap double-stranded DNA, but for hepatitis C, it contains positive sense of RNA. Right? So they are classified as the hepatitis virus because just the host cells. So they target the liver, right? So those for the genomes, they are not related by the genome, but they are related by the host cells, right? Okay, so next is the double-stranded RNA, right? The double-stranded RNA. So here, the scheme here show how the double-stranded RNA can be processed in the viral replications. So they're gonna have to be unwind first, right? To be able to make to make the mRNA. And mRNA here can go two ways as well. So first, it's gonna make like more of the negative sense of the RNA in order to make like lots of copy of itself, right? And then another route here is gonna synthesize the proteins, right? And then those proteins can be functional proteins or the structural proteins, and then they will like assembly to be the new virus. Okay, so the example of this kind of virus is going to be a rotavirus. Okay, so let's next one is the positive sense of single-stranded RNA. Okay, so this one, so you can see that the root here is a lot shorter compared to those two types of the viral genomes, right? Because it can transcribe directly, it can synthesize directly to the proteins here, right? And it has another route to synthesize the negative sense of the RNA in order to make more copy of the positive sense of the RNA. Then those positive sense can be either um, go to the viral structure assembly to be the new virus, or go back and assist the original positive sense of the genome in order to make more of the proteins here. All right, so you're gonna see that for the positive sense of the RNA for the viral genome, it's happened a lot faster, right, in the cells. So we have the example of the single stranded RNA. So we have the Picona viridae. So Picona viridae, which cause the polioles, right? And so another type is the rhinovirus. So, and then next is the Calliciviridae, which can cause the like the diarrhea, right? And when we get infected, and then the coronavirus, which is more familiar, right, for us, because everyone here know the SARS-CoV-2, right, which is belong to the coronavirus. So the name corona means the structures, right? Yeah, it means the clouds, right? Because the structures is kind of look like the cloud. So because it has the spike proteins here on the surface here. So in this class contains of the SARS. So the SARS, which emerged about 20 years ago, isn't it? And then MERS, you may heard of it, and then like the recently outbreak one, SARS-CoV-2, that actually outbreak like 3 years ago already, and um, we have like a lot of variants of SARS-CoV-2 right now. All right, so another type is the Flaviviridae, right? Flaviviridae. It contains the yellow fever, right? West Nile, Zika virus, and dengue virus, which is another important virus here that can cause like the dengue fevers, right? The dengue fevers. So those are transmitted by the mosquito. The so that's why it's kind of like outbreak often in our zone, because in our tropical zone there are a lot of mosquitoes, right? So that's why the Flaviviridae is more prevalent in our zone, right? So so you're gonna see that the Coronaviridae and the Flaviviridae are the members of virus in these families on the virus that can cause the severe disease, right? Like not much like for the chronic disease. Yeah, because one of the reason is because it contains the single-stranded RNA. Because the single-stranded RNA can transcribe directly to the proteins, right? And then the proteins can start like do some functions in the cell quickly compared to like those type of the virus, like the gap DNA or the double-stranded DNA, right? So that's why this one gonna have like more severe, can cause more severe disease and like quicker for the infections, right? Okay, so then Then there's another type that also contains the single-stranded RNA, but with the intermediate, right? With the DNA intermediate. Okay, so what it actually do for this type of virus is it's gonna reverse transcribe to DNA first. So starting from the positive sense of RNA, so it will reverse transcribe to negative sense of the DNA first, right? In order to replicate first, right? So after it replicates, so it has to be made to double-stranded DNA first, right? In order to replicate. So after replicates, right? So it can go back to positive sense of mRNA and then make the proteins, right? And another route here, they can also make the positive sense of RNA in order to like put into the viral structure, to put into the new viral structures, right? So there's also two routes here. But the most important thing for this type of the virus here, it will depends on the reverse transcriptase, right? It will need the reverse transcriptase. And in order to make minus strand of DNA and double strand of DNA. And the virus that belong to this site, that belong to this type is the human immunodeficiency virus, or HIV, right? Or and another one is like HTLV right here. So let's talk about the HIV first. So HIV, so as we know, that is the sex transmitted the virus, right? So when it get into the body, that's why like when people get infected, they're not gonna have like any symptoms. So because it will take some time, right, to produce, to make copy of themselves and then spread the infections, right, before they actually cause the disease, show the symptoms. Sometimes it will take years to be able to like to be able to show the symptoms. And another thing that it can do is for HIV is it will need the integrase. Right? Integrase is another proteins that actually integrate is from integrate, right? Integrate means like put something together. So integrase for HIV will actually function as add the viral genome or RNA or DNA into the host genome, right? So they basically add the viral DNA into the host DNA. So now the infection is permanent. So whenever that cells, right, replicate, right, like go through the cell divisions, it's gonna make the viral genome automatically, right? Even it doesn't have like the infection with the virus. So that's why it's very hard to kill. I would say it's impossible to cure the HIV infections because of integrase. So when we get infected, like the viral DNA will like go to our genomes already, and then it can also pass along to the children, right? To the babies, right? So yes, okay. So that's why it's kind of like the disease that is very like impossible to cure. Okay. So the next type is the single-stranded RNA, or the negative sense of the genome. So this one, right? So this one, because it is the negative sense, right? So it has to synthesize the positive sense first in order to convert to the proteins, right? And then like it will have two route to the second route here is to make the positive sense of RNA directly, and then that positive sense of RNA will then use as a template to make the negative sense of RNA, and then to make more of the virus here. Okay, so So the example of the virus in this in these types are, as you can see now. Okay, so it contains a lot of non-structural virus right here. So like, and there are some virus that have like this shape too. So this virus, we may familiar with Ebola virus, right? So Ebola virus will have the negative sense single-stranded RNA, right? So Ebola will cause the host to have like flu, right? Diarrhea, cold, and everything. So it was um like a hundred years ago. So I'm not sure about the numbers correctly, but the first outbreak was like 100 years ago, but it still have like an outbreak now, if especially in Africa, right? With doesn't have limited with I mean with have limited access for the proper treatment for the Ebola virus. Okay, so that is the single-stranded RNA, which is the negative sense of the negative of the single-stranded RNA. So then the next part is the segmented genome. So the segmented genome is it's kind of it's the strange genome because in the virus here it will contains the segmented of the genomes, right? They will not contain the full size of the genomes, but when they get infect into the cells, so if there are more than one type of the segmented genome, they can like combine together, right? They can combine together and make the new virus here, which contains from the genomes from both type of the genomes here. So you can say that if we start from L virus here and M virus here, so if it co-infect into the same cells, so the replication process sometimes it will put both of the genomes that originally from the different virus here into the new virus here. So that's why it can like diverse in that way. So the type of virus that like have this kind of genome is the influenza virus. So that's why the influenza virus will have high variety, right, of the variants, because it's the variants can happen very easily, right, when they just like cross with other segmented genome. So we can call this the reassortment. Okay, so this is called reassortment. Okay, so the next one is the ambisense genome. So the ambisense genome is the genome that contains the both single-stranded, both positive and negative single-stranded RNA, right? So it contains as the single chain of the RNA, but those can be identified as the positive sense and the negative sense. So I highlight the green ones as the positive sense and the olive one as the negative sense. So so it can have like a loop here in order to like to have like to increase the stability of the RNA, right? So this type of the virus will actually include the arena viridae, right? And then so this one will kind of like also transcribe to the proteins and also make copy of themselves as well, because they contain the negative sense of the RNA, right? Okay, so that is like all 7 types of the virus genome. So so I mean, like I used some example, right, of the virus. So we cannot like learn all of the existing virus. So I just show you some example of the common virus as the example of each type. Okay, so then let's talk about the viral structure. Okay, for the viral structure, it has a function, right? So the function of the structure, right? Because it has to protect the virus, to protect the viral genome, because the viral genome is very variable for the virus, because it can tell the host cells, it can hijack the host cells to make like more infections and make a more number of the virus. So that's why virus can reproduce, right, using the encoded genomes right here. So that's why it will need the protections, right? So another important thing is to deliver the genomes into the host cells, because we know that the virus will have the specific cells that it will target, right? So those proteins on the surface here will responsible to recognize the target cells. Okay, so there are basically two functions of the structural proteins. So when the virus get into the cells, you're gonna see that here, right? So this is an example of the viral entry for SARS-CoV-2. It's gonna bind to it has the spike proteins on the surface of the virus here that will bind to ACE2, right, or the cells receptor, and then they will like get into the cells like this, right, and then release the genome, right, and then after releasing the genome, so the virus can control the cells and force the cells to make copy of themselves, right? So they're gonna force the cells to make more RNA, more viral RNA, and then more of the proteins, which are the structural proteins, and then assemble to make more number of the virus. Okay, so that is the how the structural proteins can play the important roles in the viral entry mechanism. Okay, and for the structural proteins here, so it's combined of the proteins that we call the capsid proteins right here. So the capsid proteins here is the subunit of the proteins that can make it into the larger structures or structural proteins. So it's kind of like the Legos, right? So the Legos, the huge structures can be made from the smaller piece. It's similar to the structure of the virus here. It made of the multiple pieces, the small pieces, right, which is called the capsid proteins. And in the viral assembly, the capsid proteins will put together, right, in order to make the shape of the virus. Okay, and then another thing that the virus can have as the protection is the envelope here, for the envelope. So the virus will have the envelope in order to add additional protection to the viral structures, right? So some virus can contain envelope, but some virus do not have the envelope, right? So essentially depends on the type of the virus. Okay, so in order to study the structure of the virus, so as I say, we cannot visualize them by looking directly from the light microscope, right? So we will need the electron microscope. So the electron microscope will instead of using the light wave, right? So we're gonna have to generate the electron wave, because as we know that from the quantum theory that particle can be wave and wave can be particle. So that's electron. So electron can be particle and wave as well. So we're gonna make use of that properties of the electrons as a wave, right? And then to shoot at the specimen by the electron wave, and then so after that we're gonna get like the picture that derived from the electron beams, right? So cryo-EM was actually invented by like these three scientists, Duboce, Frank, and Henderson. So who got the Nobel Prize about the discovery of cryo-EMs, right? So this is basically the picture from cryo-EM on the virus. So you're going to see that this is viral structure that we can see it clearly that it will contain the structural proteins here and this genome inside the viral structures here, and maybe like the membrane proteins here as well that works as the recognition proteins. All right, so here's very clear that there's how the structure of the virus can be. Okay, so without the electron microscope, we will not be able to see or visualize the virus because it's very it's too small, right, for us to see it with the light microscope. Okay, so here's actually what happened when the virus get into the cells, right? So they're gonna synthesize the capsid protein. So after they get after they get into the cells, they're gonna make the mRNA, the positive sense of mRNA, right? And some part of mRNA will contain the code to make the capsid proteins here. And the capsid proteins is the smallest unit to make the larger structure of the virus, and then capsid proteins will be put together to be like kind of like the monomer, the polymers of the capsids here, right? And then the capsid polymers will be put together again to be like the spherical shape and like whatever its shape is, and then the viral genome will be added to these capsid proteins, and then they will like assemble to make a number of the virus here. So it's kind of like the factory, right? That makes the piece by piece of the structural virus and also with the genomes of the virus, and then put them together and then transport out of the cells. Okay, so that is what happened, and that was what virus synthesis synthesize the protons, the structural, I mean the proteins, the structural proteins. So then here is the cartoon representation of the single proteins here, the capsid proteins right here. So when they are put together to be polymers here, and then that is going to be the subunit of the viral particles here. So the viral particles is the huge particles here that contain the repeating units, right? Because we're gonna see here, this type of protein is only one or two types of proteins, and like combines together, it can make the subunits right here, and then subunits can be like repeating units, and that is the whole that can make the whole structure of the virus. Okay, so this is the representation of the virus structures. Okay, so then let's see deeply about the structure of the virus or the capsid proteins. So when capsid proteins are put together, right, they're gonna have to be the certain shape. All right, so in geometry here, so there are several shapes that that we are familiar with, right? So we may familiar with like tetrahedrons, right? So tetra is the Greek number that means 4. So tetrahedron will have 4 faces, like cube right here. So cube will have like 6 faces, right? And then how about octahedron? How many faces? 8, correct. Icosahedron is more than 10, of course. More than 12 too. More than 15. Okay, it's 20 faces. 20 faces. So and then dodecahedron is 12 faces. But for virus, the most common structure of the virus is going to be icosahedron, or 20 faces. So the 20 faces, it means that it will have like triangle here. So triangle here is the subunit, right? So there are going to be 20 triangles in this icosahedron. right? So then what virus need to make is virus need to make like the proteins that can combine into the triangle first. And then each triangle will get together and form the large structure of the virus. All right. So that will take like the numbers of proteins to make the whole virus. All right. So I actually have the symmetry rules right here. So the symmetry saying that like every subunit has to be the identical unit. So when we pay attention to this subunit, right? So this subunit needs to have the identical contact. Like if we look at this triangle too, so they're gonna have like the identity contact to their neighbors, right? To their neighbors. So those contacts will be like non-covalent interactions, or it can be like hydrogen bonds interactions, right? Electrostatic interactions, but not covalent interactions, right? Okay, so those are the symmetry rules. Okay, so here is the structure of the virus that we got from the cryo-EM, right? So as I say, right, so each triangle, so each triangle is right here, is gonna comprise of numbers of proteins. So in this example, each triangle contains of 3 proteins. So the blue proteins, the green proteins, and the red proteins here. So they can be the same types of proteins, but we classify them differently because they have the different interactions to the neighbors. Like for example, the blue proteins will have the interactions to like 4 other blue proteins like this, right? But they will have the interactions to um, four for the red or green proteins. So that's why we like classify them differently. And if we look at the icosahedral closely, and we're gonna see that there are gonna be the a few types of acids, right? A few types of acids that can show in terms of the symmetry in the structures. Like when we put the 5-fold acids here, so it can be represented by the green ones here. And if we put the acids directly into the triangle, it's going to be 3-fold acids because it is triangle, right? It's triangle. And here, if we put the acids right here at these directions, it's going to be 2-fold acids. Okay, so it's basically showing like the multiple fold acids that can be contained in the icosahedron structure. So all right, so you don't have to memorize any of this. So I just want to make sure that it's not going to be in a quiz, but I just want to show you here like how we study the symmetry of the structure of the virus. And here, from the real structure of the virus, it can represent what we model here, right? It's right. So that is the structure of the virus. So we represent the complex of them by T, right? So T means the number of the subunit that it can be present in the triangles, right? So like I'm gonna go back a little bit here. So the triangle here is our subunit in icosahedrals, right? So one here means that there are 3 proteins, 3 capsid proteins that form the single triangles, right? So you may see, right, the triangles right here. So it's contained of 3 proteins, this protein, the yellow one, the gray ones, and another yellow one, okay? And here is the structure of the real virus, and we label each proteins in the different colors. So we have like yellow, red, green, right, and blue right here. So we have like kind of like 5 different proteins. So these are another type of the virus, right, that can that can contain the like multiple subunit of the proteins. Okay, so just to know that for T1, it's going to contain 3 proteins, right? In each triangle, right? In each triangle, and the total subunit is going to be 60, right? Because if we say that for icosahedral, there are 20, right? And correct, then 20 times 3 is going to be 60, right? So to form one single virus particles for T1, they're going to need to synthesize 60 subunits of the capsid proteins. Okay, so that is T1. How about T3? So T3 will contain 3 times of the proteins that we have in in T1. So T1 has 3 capsid proteins in one triangle, right? But for T3, there will be 9. There will be 9 of them, right? So what we can see right here is each triangle is going to be this large, right? So this triangle is going to be this large. So there are like 9 proteins in total. So simply that, just like multiply 3 to 3. They're going to be 9 proteins for each triangles, right? And we have 20 triangles for the whole structure. So 20 times 20 times 9, right? It's going to be, okay, supposed to be 180 right here. So I put the wrong number. So 180, right? It's going to be 180. Okay, so let's see the next example. I think, so I have the next example right here. So these are the type of virus that have like different T, right? Like poliovirus, there's T1. Hepatitis B virus, there's T3, right? So it means that hepatitis B virus will contain 180 subunits, which is 3 times more than the poliovirus for just the structural virus, right? And then for bovine papilloma virus, we contain 7, is it T7 here? So 7 means that each triangle, it will contain 21, right? 21 subunits. And then for the whole structure, we will multiply 21 to 20, right? 21 to 20. That is the, like, how many proteins that it needs to produce in order to make just one of the virus. Okay. Okay. So next is the herpes simplex. So the herpes simplex is T16. Yeah. So it can be like thousands of units of the virus that it can make, right? Okay, so that is kind of like the complexity of the structure, right? Again, we have T1. T1 means 3 proteins in the triangle, right? 3 protein in a triangle times 60. Times 20 is going to be 60. Okay, and then for T3 is that 9 proteins, because 3 times 3 is 9, right? And 9 times 20 is going to be 180 right here. So this one is like typo for me. It's 180 instead of 120. Okay. Then here you can see that like from the subunit here. So this representation, it's maybe easier to see the pictures, right? To see the big picture on how many proteins that it need to construct the viral structures, right? So if you have T3 right here, if you count the proteins here, they're going to be 9 in total, or T4 here. So T4, if we start counting it, it's going to be 12 proteins for each phase, right? So if we have 12 proteins for each triangle face, 20 of the triangle is going to be 480, right? 480 units of the proteins, that it can be for T4. Okay, so that is the symmetry. So okay, so here's the example of adenovirus, which is one of the large virus here. It is T25. Okay, so when we like look at the structure, we will see like how complex it is and how many uh, proteins that it can contain in the single viral particle of the adenovirus. Okay, and then the second type of the viral structure is going to be the helical structure. So the helical structures will be like the rod shape right here. So inside of the structures, it will contain the RNA or DNA, right, in the helical form like this. And then the helical form of RNA or DNA will be wrapped by the protein subunit like this. Okay, so it's gonna have like the spherical shape, and then when we look at its shape under the electron microscope, we're gonna see the rod shape like this, right? We're gonna see the rod shape like this. So, so it will have like the symmetry by the rotational axis only, right? Not the icosahedral. So the this example of the virus that have this structure is the tobacco mosaic virus. Okay, so this is one of the plant virus, right? So from its names, right? So it's in fact the tobacco. So that is one type of the virus that have the helical structures. Then the next structure is the spherical structure. It has the spherical structure because it has the envelope right here that wrap the whole structure of the virus. So for the example of the virus is going to be the coronavirus. All right, so the coronavirus is one of the envelope virus that contains like the spike proteins here, right? The nucleocapsid, the membrane. So because like it can it's the membrane, the bilipid, the lipid bilayers. So it can fuse to the cell membrane in order to like release the viral genome into the cells. Okay, so that is the example of the spherical virus. And when we look under the electron microscope, we're gonna see the structure here, with like spherical shape, right? With the spike protein. Okay, and then the last one here is the complex structure. So this one is called the bacteriophage. The bacteriophage is the virus that infect the bacteria, right? So it's the virus that have like very interesting structures. So it has the icosahedral head that contains the nucleic acid or the DNA, and then it has like a neck right here, and then it has like the like legs right here. So when it get infected to the bacteria, it will inject the genomes right here to the through the membrane of the cells to the cytoplasm of the infected cells. Okay, so we're gonna be looking at more on the viral life cycle after this. Okay, so viral life cycle. Okay, viral life cycle is the process of the virus when get into the cells, what it does here, and then like how it's assemble and make copy of themselves and then release outside of the cell. All right. So this one is the example of SARS-CoV protein, SARS-CoV virus when they get infected into the cells. So they will bind to H2, right, for on the cell's surface here, and then it will be like transport into the cells by endocytosis, and then at some point here, it's gonna fuse its viral membrane to the membrane of the cells right here. I mean of the envelope here as well. So it will then release the genome, right? Then release the genome, which is the RNA, the positive sense of RNA that can be replicated and then uh, transcribed to the proteins to make more structural proteins or non-structural proteins, and then go through the endoplasmic reticulum and then go through Golgi apparatus and then combine or assembly with the structural proteins here to make the new particle of the virus here. So like this side is the MERS code. So they work like similarly. So that is basically what happened when we get virus into the cells, and um, that's pretty similar to all of the virus, right? But this one, I just used the coronavirus as an example. But if we have like different types of the genomes, it's gonna have to add like the steps right here, because here we have the mRNA, right? The positive sense as the genome, it can like transcribe to protein directly. But if we have the DNA, so DNA has to be made to mRNA first, and then mRNA can make the protein. So it's going to add more steps right here in the replication and transcriptions. Okay, so that is the viral life cycle. But here for the bacteriophage that I showed you previously, so bacteriophage is gonna attach to the host cells right here, and then like a needles. So the genome of the phage will be added into the cells, right? So you can see it like this. It's kind of like the needles that puncture through to the cell's membrane and then release inside the cytoplasms here, and then it can start working right away. So it can start making copies of more bacteriophage right here, and then assembly, and then like break the cells to be able to release to more of the phage to spread the infections. So this is what happened when bacteria get infected. Okay, so bacteriophage is very interesting because it cannot like I mean as far as we know right now, it's not gonna be harmful much for our bodies, right? So sometimes people use it to for the treatment of the bacterial infections. For example, if the bacteria is resistant for the antibiotics, right? So they're gonna be like a phage development for those kind of bacteria, right? In order to just like get those bacteria infected, and then they will like burst, right? And then we will able to remove that bacteria. So we call those phage cocktail, right? So you can like try to if you're interested, you can like Google it and look more about this kind of phage. And then because like you learned with Ajahn Mike, right, earlier. So I think he mentioned this a little bit because he has some project that are working on the bacteriophage as well. Okay, so that very interesting story. And bacteriophage is also classified as the virus as well, because it contains only the DNA or RNA, right, which is very important for the virus right here. So it falls to the category of the virus, which cannot have the cells have the activities by itself without the host cells. Okay, so then we will move on to the last part of the lectures, which is the antivirals and the vaccines, right? So we learned so far that what virus are gonna do to our body, and then how do we prevent it, or how do we fight against those infections, right? So there are two main type of the treatment. So the first thing is we call the antivirals. So antivirals will like act directly to the virus in order to stop or slow down the viral replications or viral entry. So basically, you can like design the inhibitors, right, to block in any steps or multiple steps in the virus. So for example, here, so we have the SARS-CoV-2 again virus that trying to get into the cells, right? So we know that it has to bind to ACE2 using its spike protein. So we may design the small molecules or any type of biomolecules that can block the interactions between ACE2 and it's the spike proteins here in order to stop the entry mechanism. And next is we can mess with the viral replications here because we know that the virus will contain the non-structural proteins, which is the functional proteins like polymerase, protease, helicase, right? So we can block the activity of those proteins by designing the inhibitors that will deactivate those proteins, right? Or block and at the active site of those proteins, right? And then using that method, we can slow down of the viral replications and then even kill the virus and cure the infected cells, right? So so that is how we design the antivirus. And another thing we can also do is we can also block at the host cells proteins because virus cannot express everything that they need in order to make copy of themselves, right? Some proteins they're gonna make use of the host proteins. Like some of the virus will make use of the host polymerase or host protease. So if we can like block those proteins and the viral replication can be slowed down as well. But the side effect can be occur as well, right? If we mess with the host protein. So that is kind of trade-off of designing the inhibitors or the antivirals. So the antivirals doesn't have to be just the small molecules of drug. So they can be like the peptide as well. So they can be like a huge protein as well, as long as they do the function in order to stop the viral replication. So they will, we will call them the antivirals. Okay, so here is here are the example of the inhibitors of the polymerase here. So for the polymerase, we know that it's the process that genome will be made, right? The multiple genome will be made. So if we block these steps, right? So no more of the viral genome will be made. So we can stop the replication by stop at the viral DNA polymerase or viral RNA polymerase as well. So these are example of the drugs that developed and approved to use against the viral polymerase, right? So this one is the sofosbuvir, right? So you're gonna see the structure right here. Favipiravir. So we often heard like when the outbreak was happening. So some people take the favipiravir, right, for the antiviral against the SARS-CoV-2. So here is the last one is the Ranvir, right? So which is the another small molecules that can block the polymerase. So you can see those are the nucleotide analogs, right? So the nucleotide is the building block of the proteins, right? Is the building block of the I mean not the protein of the DNA or RNA, right? Because of that, this can be recognizable to the polymerase, to the DNA polymerase or RNA polymerase, because the polymerase if they need to make the new copy of the DNA or RNA, they have to be attached to the DNA or RNA chain, right? So if we have the small molecules that mimic the structure or the subunit of DNA or RNA, we can actually block the active site of the polymerase too. So that is how we designed it. All right, and then we're gonna have to like further design it a little bit in order to like have it like more more attached to the proteins. Okay, so this is how like more like theory. So you don't have to memorize any of this. So I just want to show you that it's related to delta G, right? So delta G. So in thermodynamics class, we know that delta G is if it's less than zero, it's gonna be favorable, right? So when we design the inhibitors, if we can like calculate for the delta G and find that delta G is less than zero, it's going to be favored and it can be a good inhibitors. All right, so that is the help for the design. Okay, so this is example. If we have like more magnitude of the negative value of delta G, like the stronger of the inhibitors and the better drug is going to be, right? So compared to like this orange drug, the red one is thicker, harder than the protein. So it's more efficient in order to be the inhibitors. Okay. So so then next is the vaccines. So unlike the antivirals, right, that stop the viral replication directly, but in vaccine, we pay more attention to our immune system, right? So basically, for vaccine, we're gonna have to add part of the virus into the bodies, right? And then allow the body to recognize that that pieces of the virus, and then allow them to make the to alert the immune system, right? And then for the next real infection, we will have like the antibody, right? The antibody that can recognize on the virus. So basically, if we want to recognize the SARS-CoV-2, for example, for the vaccine, we might add the spike proteins into the vaccines, right? So when the spike proteins get into the cells, so the cells will see and look at it as the particles, right? The invaders, right? The invaders. So they will trying to find a way to remove it, right? So when we have the real virus, that will contain the spike protein as well. So now the body is not to don't need to learn, right, about how they're gonna neutralize the new invaders, because they have the memory that if they have this kind of invaders, how they're gonna use for the antibodies, right? So that will help the body kind of like pre-learn about the structure of the invaders. So that is the use of the vaccine. So basically, boost our immune system prior prior real infections, right? So the virus, that's why the virus um, that we use in the vaccine needs to be made them weaker first, right? So many types of the vaccine will modify the virus to be weaker forms, right? And then inject to the body. So allow the body to learn how to fight against them in the weaker forms. And then for the next infection, if we have like the stronger form of the virus, so we know how to fight it efficiently. So it will like help us boost the immune system. And more modern type of the virus, type of the vaccines, right? So we have like the RNA or the subunit vaccines, which is the vaccines that break down like some important part of the virus and kind of like injected to our body, right? And then for RNA, right? For RNA vaccine, because we know that RNA can transcribe to the proteins directly. So if RNA can encode the proteins of the viral proteins, and then we inject that RNA to the body into the cells, so the cells can learn how to recognize that proteins and how to fight against that vaccine before the real infection. So those how the vaccine works, right? Depending on how do we want to train the cells, train the immune cells, right? So that's why there are multiple types of the vaccines right here, right? So the vaccines will basically help us to boost the immune system, right? And also the real infections um, also alert our immune system too. But the side effect of the infections is you're gonna have the symptoms, right? So you're gonna have like, for example, if we get infected by SARS-CoV-2, we might get like the cough, right? The severe cough and the inflammatory in lung, and then like many symptoms that can caused by COVID. But for vaccine, we might get a like fever for just 1 or 2 days, and that's it. So that is how we make use of the vaccine. Okay, so I think we have about like 20 minutes for the quiz. So do anyone have any questions? Yes. Rectangular? Yes. Yes. So you mean if it's cube instead of the icosahedron? So if it's cube here, yes, it's not gonna be equal to 20 times, right? Because icosahedron, we know that there are 20 faces, and each triangle will contain up 3 proteins, right? So 3 times 20 will be 60. But if you have like a cube structure here, so if each face of the cube contains of 3 proteins, the same. So 3 times 6 is going to be only 18 proteins that actually need to make the whole structure of the cube. So that is how different. But those are not common. But I mean, like the common structure of the virus is icosahedron. So that's why like this number is kind of like more general for icosahedron. Yeah, icosahedron. Yes, yes. No, not many, not many. So yes, only icosahedron. So here, yeah, but for the general shape of like everything else, we're more familiar with uh, tetrahedron, cube, or octahedron more, right? But for virus, it's gonna be like more common in icosahedron more. Okay, so that is very interesting for the structures, right? So yeah, so this is how virus is, and like um the advancement of the electron microscope like reveals us more about like how interesting natures are. So that's what we found from the cryo-EM, like this kind of structure. Okay. All right. Any other questions? Yes. Yeah. You mean non-structural proteins? So, okay. So the question is, so let me rephrase it. You mean the structural proteins and what the difference between the structural and non-structural proteins? Or oh, okay. Non-structural proteins and structural proteins, right? So the structural proteins are the proteins that will be included in the viral structures. So they can be capsid proteins, or they can be like spike proteins or envelope proteins. So basically, if we break down the virus, right, we're gonna see these proteins as proteins, right? But for non-structural proteins, they are not the part of the virus particles. They will be transcribed and just work to assist the virus into the replication process only, right? Not then they are not packaged into the virus. Yeah. So that's why when we break it down, we're not going to see the proteins like these non-structural proteins, but they of course they have the code here, right? They have the code to make those proteins in the viral genome. Yeah, so that's what difference between the structural and non-structural proteins. Okay, there are only two types of the proteins here. Yeah, no problem. Yes. Okay, yes. 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 Okay. Thank you. So let me clarify a little bit. So we when we have the DNA, right? We have two route for the mRNA. So the first groups of mRNA will be made, and then the protein will be made out of these groups of mRNA, and then not only the structural proteins can be made, right? The non-structural protein can be made as well, right? So these will transcribe to all of the proteins that encoded into the viral genome. But this one is made for copy to make more copies of itself, right? Because in order to make the copy of themselves, they need to make a lot of DNA, right? And then they're trying to make a mRNA first in order to make the negative the negative strand of the DNA. Then the negative strand of the DNA will be combined to the positive strand of DNA in order to make like the full structure of the genome, right? Then ready to be packaged and assembled with the structural proteins there. Okay. All right. So that are two routes that we have. Okay. So for the students from WebEx, do you have any questions?
How do we find a way to cope with new viruses?
Can you say it again?
Like, how do we find a way to cope with new viruses?
How do we find a way to cope with new virus, right? Can you type? Can you type? Because I think the the speaker is like it's kind of like not clear for me. So can you type your questions into the chat box? Oh, here. Okay. I'll cope. How to cope. Okay, I'm sorry. Okay, so cope is like how to deal with the new virus, right? So the first thing is we have to study the nature of the virus first. So we have to classify the virus like maybe by their genome. So whether they are DNA type virus, whether they are RNA type of the virus, then we will kind of know like the likely behavior that the virus will be. Then from that understandings, right? So we can design the new inhibitors or the the new vaccine based on the understanding of those viruses, because the different genomes of the virus will require the different types of proteins, right? So for example, we might not require to have um, transcriptase, I mean reverse transcriptase for double-stranded DNA, but we do require to have the reverse transcriptase in positive sense or negative sense of RNA, right? For example, like that. So then after that, we can identify the functional proteins of the virus or the proteins that the virus need, right? And then after that, we may be able to design the new inhibitors, right, against that virus, and to design the vaccine that can heal. I mean, that can protect the virus too. So the antiviral is for the treatment for cure, right? But for the vaccine is more like for the protection, right? And then another thing is the good thing, the good answers, like an easiest answer is try not to get infected with the virus, right? Or or stay away with all the risks that you might have, right? For the virus. Yeah, that is like immediate response that you can do, right? Because the development of the antivirals and for the vaccine takes time. So yeah, the immediate ones should be, yeah. And again, and you may like protect yourself by stay healthy, right? Exercise and sleep enough, sleep well. Yeah, that's another thing that you can like do, because the healthy people and the weaker people will respond differently when they get infected with the new virus. Okay. All right. Thank you so much for your questions.
You.
All right. Do you have any other questions here? Okay, so if not, actually, I have a quiz here, but we don't have enough time to complete within a class, so I just then maybe assign as a homework. How about that? So actually, I do have the link right here. So the quiz contains 25 questions of the multiple choices. So so basically, I will like give you about 45 minutes to complete it, but based on your performance last time, you just need like 15 minutes to complete it, right? So so I want you to like try to practice as a homework, all right? So then um after everyone submitted, so in the, I mean, the earlier of the next week, so I'm gonna release all the answers and the solutions. So about the quiz last time, I think we have the new students, right? So I'm gonna have to check if like whether she completed the the quizzes or not. So then if everyone completed, so I'm gonna release it like the same times of this quiz. I mean the solutions. I'm gonna release the solution and answer for this one as well. Okay, so this one hopefully is not like too hard for you um, because I don't want to make it to be like heavy virology class, but this one is the general class, right? So okay, all right, that's all. And then if you have any further questions, you can email it to me, or I'll be happy to discuss. All right. Thank you so much for today. ครับโอเคครับ Oh, the deal. I would say within this Friday would be like ideal. Friday, yeah. Uh, I mean, like Friday to be specific. Friday 11:59 p.m. Okay, but I mean, like if you have time, like better to finish it earlier, right? Because like you still fresh with everything you have learned so far, so it's better to start it quickly. Mm. All right. Okay. So thank you so much, everyone, for your attention today, and have a good day. So we're gonna meet again. We're gonna meet again next week, right? I guess Tuesday. Okay.